Hello again. We're going to continue our go from a gene on DNA to a protein that could result in a trait or should result in a trait for an organism. So in the last video clip, we talked about how there are two processes associated with that. And unfortunately, they both sound kind of similar in terms of their names. One is transcription and one is translation. And if you go back and take a look at the material we've already covered, the process of transcription is where a gene on DNA is copied onto a single-stranded RNA molecule. All right, again, that process is going to be called transcription. And now transcription occurs for any of the types of RNA that we discussed in the previous lecture. So there are ribosomal RNAs, there are tRNAs, there are these messenger RNAs. We're really focusing here, though, on thinking about messenger RNAs for the time being, because those are the ones that are going to go to a ribosome, and the ribosome will actually read the sequence of the A's and the G's and the C's and the U's on there, and using that information, amino acids will be linked together to build our protein. So transcription is going to be our first major process here. And we're taking a look at this in prokaryotes. And keep that in mind. So prokaryotes, like bacteria, eukaryotes include some additional steps with transcription before the next phase, translation, the protein synthesis phase, can take place. And just to kind of trim down the material for this course, we're not going to go through some of those events that eukaryotes do that prokaryotes don't. That gets into introns and exons and splicing. So if you're reading about that in your textbook, that's what I'm alluding to here, that we're not going to wind up covering that in this particular class. All right, transcription. So let's take a look at this diagram. We've got this purple molecule here. That's supposed to be a double-stranded DNA molecule. So we're not seeing the A's and the G's and the C's and the T's written in. And I've got highlighted here that this stretch of the DNA is a gene that codes for a protein. All right, and notice here we've got this brown blob, RNA polymerase. So with transcription, we're going from DNA to RNA, correct? Right. Which means we're going to build an RNA molecule. So what enzyme is going to do that? RNA polymerase. Remember, we had DNA polymerase enzymes that build DNA molecules, so we have RNA polymerase enzymes that build RNA molecules. So the RNA is going to do the actual transcription process. Now, we don't want RNA polymerases just randomly doing transcription anywhere on a piece of DNA. It needs to only do transcription of a gene, this particular stretch of DNA. So that means the RNA polymerase has to know where to start this process. So here we've got our gene on DNA, and upstream of there, notice they're highlighted in yellow. There is a stretch of DNA that's called the promoter, and that stands for promoting transcription, attracting in the RNA polymerase, basically waving a red flag and saying, hey, you need to attach here and start the transcription process. So we sometimes say the start location for transcription is just beyond the promoter. The promoter is going to attract in this RNA polymerase enzyme, and then it's going to start copying that stretch of DNA that we're calling a gene. So if you look at the rest of the diagram here, that RNA polymerase enzyme is going to start sliding through the double-stranded DNA, and it's going to open up the double-stranded DNA, kind of like we saw with DNA replication and that helicase enzyme. The RNA polymerase is going to temporarily peel apart the DNA. And then here what's going on is the RNA polymerase is going to read one of the two DNA strands. That's called the template DNA strand. And if it sees a G, like they're showing you here, if it sees a G on the DNA strand that's purple, it's going to do complementary base pairing. It's going to bring in a C nucleotide for our messenger RNA that we're making. When it sees a T on the template strand of DNA, it's going to put an A in the messenger RNA copy that we're making. If it sees a C, it's going to put in a G. If it sees an A, what is it going to put in? It's not going to put in a T it's going to put in a U on the messenger RNA that we're building. So in this diagram here in orange, or reddish orange there, that's the new messenger RNA molecule that's being built. And we can call that a messenger RNA, or we can call it an RNA transcript. Messenger RNA is often abbreviated with a lowercase m and followed by RNA. All right, and then also we need to know where to stop this transcription process. We only want to copy the DNA, the actual part of that's a gene, and then we need to stop at the end of that gene. We need to stop the copying, and that occurs at a location highlighted here in orange that's called the terminator. So basically, kind of in a nutshell, the RNA polymerase is going to fall off the DNA at that point, and then your messenger RNA or RNA transcript is released from the DNA. All right, and this can happen over and over and over again. So you can have more RNA polymerase enzymes here that attach, transcribe, transcribe, transcribe. And so typically you're going to make many, many, many messenger RNA copies of a particular gene that you are expressing in this particular cell. So that, in a nutshell, is transcription. And again, if you're reading along in your book, which hopefully you are, in eukaryotes, before we can move on to the next step, which is translation, there are pieces of the messenger RNA that get clipped out, and then the parts that remain get fused together. That's called messenger RNA splicing, and the parts that get clipped out are called introns. The parts that remain are called exons. And so you'll read a little bit about that in your text, but I'm not going to require that information for this particular course. All right, so once you have made your messenger RNA copy, then we are ready to do protein synthesis, because remember, we're going from a gene on DNA to this messenger RNA copy. That's going to provide the instructions for how to build a polypeptide that will fold up into a protein. And then your proteins are responsible for phenotype or traits that an organism has. So this process right here of going from messenger RNA to polypeptide or a protein is called translation. And again, you are changing languages. You're going from the nucleotide language of the DNA and the messenger RNA to the amino acid language of proteins. And so that's where that term comes from. Where is this occurring? This is occurring on the ribosomes. So we've already talked about that before in this class. There's another type of RNA that gets involved in this process, and those are called tRNA molecules. And here's one with kind of a three-dimensional look. You'll see these drawn or sketched out in different ways. But tRNAs are molecules. They're kind of like factory workers that are going to bring in the amino acids that are going to be linked together to build a protein. So one thing I want to point out here, this is our polypeptide or our protein: methionine, arginine, tyrosine, leucine. Those are all examples of amino acids. Most cells use 20 different kinds of amino acids to build proteins. But those are names, and you would see those on the back if you were taking some sort of protein or amino acid supplement. You would see amino acid names like that on the ingredient list for that supplement. So what tRNAs do is they bring in these amino acids and they match them up. They match up the amino acids with a series of 3 bases. Remember when we're talking about the A's and the T's and the G's and the C's on DNA, or your AUGC on RNA, those are called bases for nitrogen-containing base or nitrogenous base. The tRNAs are going to match up these amino acids with stretches of 3 bases on the messenger RNA. And those 3 bases are called codons. And that's the basis for our genetic code that we use, or all cellular living things that we know about, use to go from the DNA, RNA language to the protein language of amino acids. All right, and so what this is called a codon table. And you will see many different versions of these. The one in your textbook looks a little bit different. And so as I mentioned, back over here, on a messenger RNA molecule, a series of 3 bases together functions as a codon specifying that a particular amino acid needs to be included in a protein that we're building. So if we look at this table closely, take a look at one of these closely. You can look at the one in your book if the one here on the screen is not visible. This is basically a chart which lets you know, okay, if on a messenger RNA I have these 3 bases, what amino acid is that specifying? So for example, right here, if you have UUU on messenger RNA, that specifies an amino acid called phenylalanine, which is abbreviated with PHE. Or right here, AUU specifies in a protein an amino acid called isoleucine, which is abbreviated ILE. And so there are 64 different combinations of RNA bases that you can have, 64 possibilities, and there are 20 different amino acid possibilities in most cell types. And so another thing that's telling us is that there are individual amino acids, more than one codon can specify those. And if you look at the table closely, that will be pretty obvious to you. For example, all 4 of those codons, CCU, CCC, CCA, CCG, if those occur in a messenger RNA, that is going to specify that a proline gets included in the protein that's being built. All right, some special codons we have right here. AUG is a special one. That is called our start codon. So just like you have a start and stop location for DNA replication, remember that? We had our origin and our terminator. We just saw that there's a start and stop location for transcription, promoter, and terminator. You got to have a start and stop location for translation as well. So AUG is the first codon that gets used when we're building, or gets read when we're building a protein. And that codes for an amino acid known as methionine. That's in eukaryotes. In prokaryotes, there's a slightly modified version of that amino acid called N4-methionine in bacteria. All right, and then finally, stop codons. You've also got to have a place where we know we've got to stop translation, stop building the protein. And so during translation, when a UAA, a UAG, or a UGA is encountered, that's going to stop the protein synthesis process. So for right now, we just want to develop a little bit of familiarity with this table. As we continue to move through this story, we're going to see in more detail how this genetic code works and how we go from a messenger RNA copy of a gene that was made during transcription, and how we go from that to having a sequence of amino acids bonded together that can fold up into a 3-dimensional protein. So we will continue that story with part 3 of transcription and translation.